My name's Simon Gregory. I'm an Associate Professor at Duke University in the Centre for Human Genetics. The reason I got into MS research was I had finished my PhD in the UK working on the Human Genome Project, and I joined the Duke Centre for Human Genetics, and they wanted someone who had sort of a molecular background and a genomics background to work on an existing MS grant that had been awarded to Jonathan Haynes at Vanderbilt. And he was collaborating with our former centre director here at Duke, and they needed someone with my expertise to be able to contribute. So the work I'm doing on MS is it has multiple different directions. The primary grant, or the direction that I'm working on at the moment, is to figure out how interleukin-7 receptor has a role to play in the development of MS. So several years ago, in 2007, we published Genetic Association of IL-7R, and it was the first gene for something like 20 or 30 years that had been identified as being genetically associated with the disorder. And so we wanted to figure out, it was okay to identify the gene, but really my interest is what does the gene do and how is it implicated in the development of MS. So what we did was submitted grants to the NIH and grants to the MS Society, and both of them were funded. And we're using that money to, first of all, figure out how different sort of versions of the same gene. So what we found is a single nucleotide variant places individuals more at risk than individuals who don't have that variant, risk of developing MS. So what we want to do is say, all right, in MS patients, how does that single nucleotide variant influence T-cell signaling, so immune cells that are important in the development of multiple sclerosis. We're also trying to identify what are the mechanisms that regulate the production of two different versions of that gene, because we think that potentially if we can identify that mechanism, those proteins that regulate the production of the soluble version of the gene or the receptor version of the gene might be themselves candidates for developing MS. So that's that particular project. We're also involved with looking at again, immune cell metabolism in the context of the gene that we identified, because we think there may be something to do with immune-specific T-cell metabolism based on the DNA variant we identified in IL-7R. We also are making an IL-7R mouse, so through philanthropic donation from Herman Stone, we're making an MS mouse that has replicates what happens in the human, because the mouse has different sort of setup of IL-7R. We want to sort of humanize the mouse and make it produce the human copies of the gene that we're interested in. Down in Kannapolis, we're working with the Murdoch study. So Mr. Murdoch funded the Murdoch study, which is sort of a cross-sectional study collection of as many people from the Cabarrus and Kannapolis counties as they can collect. And as part of that study, we're also collecting MS patients through collaboration with Carolina's health system and various neurology practices around the area, and also extending down to Greenville in South Carolina with Mary Hughes. So what we want to do there is collect 1,000 MS patients of all different sorts, so relapsing, remitting, secondary progressive and primary progressive MS, and then use those samples, those 1,000 patients, as the basis for identifying biomarkers that are associated with either development or the progression of the disease. At the moment we've got around 350, and we're just about to start our first study looking at individuals who have MS but aren't on drugs, and compare them to control individuals from the rest of the Murdoch study. What we're going to do is look at their metabolites in their blood to see if we can identify anything that's different in the sort of chronic inflammation process associated with MS. And just recently we've agreed that we will also collect serial primary progressive MS patients. So the PPMS is a definitely underserved part of the MS community, and what we want to do is collect biannual samples of our primary progressive MS patients, and so what we'll do is collect them twice a year and then do various sort of profiling, use various profiling platforms to figure out what's associated with their disease and what seems to be developing during progression. We want to collect, these biannual samples for at least 5 years. At the moment we only have 16 primary progressive MS patients. We'd love to get more, and so we're trying to increase our cohort and study those sort of aspects of the disease development as well. We identified IL-7R or published IL-7R in 2007, and since that time there's probably another 50 genes that have been identified. And the difference is it's really the availability of technologies that allow you to screen literally millions of markers across the genome at once and tens of thousands of individuals. And so what you had is the International MS Genetics Consortium coming together with, it started off with 6 groups and it's got more than 20 now, and they screened something like 10,000 plus patients to identify genetic susceptibility or risk alleles in the genome. And so what they identified is, tremendous, either validated what we'd, the handful that we'd identified before, or identified, dozens of others. Now the difficulty is with that approach is that these variants are present in everyone. So, for example, the IL-7R variant we identified isn't present only in MS patients, it's present in non-MS patients as well. It's just that they more frequently have these variants than people who don't have MS. So the difficulty is trying to figure out why, if they're in normal people or who don't have the disease, why is it they have higher incidence or higher frequencies in MS patients. So the 52 genes that we have at the moment, these are common variants which probably contribute to the development of MS, but they're also present in the population, and so people are analysing and say, alright, if it doesn't take one variant, is it 5 variants or 10 variants, but it increases your risk of developing the disease but doesn't cause the disease, because as we know, common diseases are caused by, common complex diseases are caused by an interaction of the genes we're born with and our environment. So potentially you have a background of genetic susceptibility, and then you have a bacterial or viral or potentially vitamin D deficiency that may overlay on top of that genetic background and develop the disease itself. The population that we screened were Caucasian, and it raises a good point that not all diseases express themselves in all different races. What we're relying on is the patients, the Caucasians or the white patients that we primarily investigated, because they occupy the largest section of the MS community, if you like. We looked at individuals who have a Caucasian ancestry, so they come from Northern Europe. So they have this sort of genetic background that derives from those sort of regions that are genetically susceptible. But having said that, people have looked at MS patients in Sardinia and in Italy and more northern climates. So it's sort of there's this concept that, and it's you look at the epidemiological analysis and there is increased incidence away from the equator and towards the poles. But I think we're sort of capturing those individuals because we're looking at Caucasian, I suppose, genetic background in the people that we're looking at. I think more epidemiologically, is there less sunshine? Are there more colds, more viral infections and all that sort of thing in those places? But it's on top of the genetic background. At the moment we're not looking at differences in gender. We're sort of, in the cohort here, we're seeing most of our patients are females that were, as you expect in MS. In the MS patients we're recruiting at Duke, where we're looking at IL-7R, most are females, and what we want to do there is really match them with same-sex controls of a similar age, so we're hopefully controlling for that. In our 350 that we're steadily trying to achieve our 1,000, most of them are females as well. We haven't got to the point at which we have enough numbers to say, alright, in relapsing remitting or secondary progressive, once you start subdividing the population by age or sex or race, you start getting down to smaller and smaller numbers. So that's why we really want to increase it to 1,000 to be able to say, all right, let's try and question these, really interesting observations about, aggressive disease or sex-specific disease or the different types of disease. I think because we're not just using one approach to attack the disease, we're using multiple different sort of approaches where, like I said, we're developing the mouse, which will help us hopefully develop a better model than EAE, a more genetic-based model of MS to help us understand what the role of IL-7R is in the development of the disease using a mouse model. In the humans, we want to identify, what pathways may be implicated, and we still don't know whether it's the signaling of this particular gene in our immune cells or it's the effect that it has on the stability of the receptor, this gene in the cells itself to help mediate signaling. We've got some preliminary data which seems to suggest that our hypothesis that the proteins that regulate the production of two different versions of IL-7R may be associated with MS. So we're identifying, using a different approach to find more candidates and sort of flesh out the pathways. And our Murdoch MS population really is an exciting population because we're collecting samples for blood, for RNA, DNA. We're going to be able to use serum and plasma proteomics and metabolomics. We're also looking at urine. And that spectrum of samples will allow us to sort of ask very different hypotheses. I was talking to someone here at the Duke Center of Human Genetics yesterday about a field of study which I hadn't been aware of before, and we'll probably look at the urine of MS patients to look at endosomes. So these are exosomes, sorry, which are a cell-free DNA and very small gene expression products. So I think, for my sake, if we make one step forward on any of those projects, it makes me very happy. Mm-hmm. If we make multiple discoveries, like what's associated with disease development or what's associated with progression, like we'll hopefully see in our PPMS patients, then we have potential to figure out what are the mechanisms that are driving progression, and can we identify a gene or pathways that are associated with progression that we can stop, which prevents the progression of that that horrible disease, or can we translate into, say, something like secondary progressive MS? Is there similar mechanisms in SPMS that we can use to help those parts of the patient cohort as well? My research is really it's not focused on a biomarker for development or a drug for prevent progression. It's sort of trying to understand the whole pathogenesis of the disease, which will enable us to answer, do multiple things. Develop an assay for screening. I know a lot of the MS patients I talk to are very concerned about their families because it is a genetically inherited disease. Can we develop a screening tool? If we understand the pathways, can we develop a drug that is going to stop the pathway, the progression of the disease based on the genes in the pathway? It would be great to develop, a biomarker of, say something like if someone is in remission and they pee in a cup, and something like a test that you stick in the urine to say, all right, there's a relapse coming or something like that. So I think it's multiple different things, not just to focus on one particular aspect. And ultimately, it's all for a cure. I've given multiple talks to MS, National MS Society advocacy groups over the years, and we, as scientists, we can't do any of this work without their input and their contributions, not only donations, but actually giving blood and urine and all this sort of stuff. But really, it's all to benefit them, because sometimes science can be self-effacing. You're only finding out things, for your own sake and self-fulfilling. But I think when you have diseases like MS, where it has a tremendous impact on the person and the family, extended family, it's really, really important that you don't lose focus of what it is that you're doing, and that you are trying to find a cure or slow the disease down or start moving into primary progressive MS, which is very underserved, as I said. So I don't think you should limit yourself to any particular aspect, but ultimately it's the cure, because no one wants the disease. The thing about human disease is that no one knows everything, and even when you get MS researchers in a room, MS as a disease affects so many different potential mechanisms that you can have a room full of MS specialists, whether from clinical through basic science, and there's just so many different things you can talk about. It's an intriguing disease, and you're never short of potential things to try or hypotheses to try and answer. And as a researcher, you want to be challenged on multiple different levels, and MS is definitely a disease that does that, because it's very complex. And it's certainly taking me into fields like the mouse. I've never done mouse work before, and it's taking me into mouse, which should be really exciting. We're hoping that we have a mouse with something that develops MS, or we challenge it and may develop MS. The genes that we think may regulate IL-7R, if they are genetically associated, it's a completely different way of trying to find association, rather than just taking 10,000 people and saying, all right, what is implicated? We're looking at sort of a different approach of saying, all right, what are the mechanisms that regulate some of our known candidates, and can those mechanisms also be important? And working with a medical fellow here, Thad Kuntz, who's very keen on collecting CSF from MS patients, and he's very interested in looking at gene expression in CSF. I think that's tremendous, and if I hadn't been working on MS, I wouldn't have met with him and had these biological samples to start interrogating. So it's sort of a network of different things that are exciting, and different approaches and different opportunities. But they all have to funnel down to finding a cure. If someone was newly diagnosed with MS, I'd tell them not to think the worst. Invariably, people will go to the internet and find the worst case scenario. I'd tell them they're not alone. The MS Society does an excellent job of helping people understand what the disease is, helping them tie into patient advocacy groups, local ones or nationally. I'd tell them to be informed. I'd tell them to make sure they understand that it isn't, a death sentence. Drugs have been developed. Having spoken to a lot of MS patients, even just having an oral pill that they can take was a huge step forward. Myself, I know I don't like injections, and I can't imagine, having to do that on a daily basis sort of thing. And I'd tell them not to give up hope. I think hope's a very important thing, and I think everyone deals with sort of adversity differently. And I know MS patients who would prefer not to be in groups. They deal with it on their own, but I also talk to patients who enjoy the camaraderie of people who can help you understand, expect what's expected type thing. But I'd say, don't give up hope.